Purification of water from carcinogenic heavy metal contaminants to sulfidation and ultrasound and its effects on Girardia, Tigrina, and Lemna minor. In this era, many people focus solely on success and pay little attention to the rules that they're breaking and the effects of their actions. And oftentimes, their actions have a massive impact on the environment and ultimately all of us. For instance, carcinogenic heavy metals like mercury, lead, and chromium have been found in lakes throughout America at levels that have been proved to be unsafe. And that brings me to the purpose of this project, which is to test the viability of sulfidation and ultrasound in the detoxification of heavy metals in drinking water. In addition, this project will be comparing the effectiveness of both of these processes and looking at all the adverse effects that it has on aquatic life. Antibiotics, psychoactive drugs, hormones, and other carcinogenic heavy metals, and many other substances enter the water cycle and the food chain without people being aware of it. The processes that are currently being used in the sewage treatment plants do not completely break down these biologically active substances. So a method that has been proposed to solve this problem is sulfidation, which is the process of installing sulfide ions in a material or molecule. The process is widely used to convert oxides to sulfides, creating a less toxic compound. But why should we use more chemicals to remove the existing chemicals? So recently, a new way to purify water without resorting to chemicals has emerged, which is ultrasound. This process combines an extremely high-pitched sound with high pressure to create thousands of tiny cavities in the water. They decay spontaneously, generating so much energy that it is capable of breaking down all impurities. Entities. Well, my first entity is planaria, which are tiny free-living freshwater flatworms. Their remarkable ability to regenerate themselves enables planarians to replace parts of the body removed by amputation or naturally occurring fission, and also makes planarians an interesting entity to study. The regeneration relies on a large population of stem cells called neoblasts. And recent research has suggested that a lot of the important transcription factors found in human stem cells are also required for function in the planarian neoblasts. Stem cells are very important for human biology and vital life processes like growth, wound healing, and aging, just to name a few. And because of the selective response of stem cells to stimuli, planarians could be used experimentally to make a statement about the effects of certain chemicals on stem cell function across the animal kingdom. The next entity is Lemna minor, commonly known as duckweed. They're extremely small, free-floating aquatic plants that grow in dense colonies on the surface of water. Lemna minor serve as an easy-to-use entity, as one simply has to count how many fronds developed over a 5-day period, which will depict the sensitivity of duckweed to the particular chemical. Experimental design. Well, obviously my first step is going to be to prepare the solutions. The sulfidation process is pretty easy. It's literally exposing the solution to a sulfide ion. Well, after that, I'll be performing 4 different tests on the purified solution. In order to test the concentration of lead and chromium ions in the solution, I'll be using the GCMS. Then I will directly test for toxicity by exposing the solution to E. coli and seeing what happens. And as I've mentioned before, in order to test the effects of sulfidation in aquatic animals, I'm going to be using planaria, and after 7 days of complete exposure to these chemicals, I'll be looking for 4 different things. First, how long they took to regenerate. Second, the size of the newly grown planaria. Third is the observational regeneration assay, which has to do with the reappearance of photoreceptors. And fourth is the functional regeneration assay, in which I'll measure the sensitivity to light and the PR appearance. Now, for aquatic plants, I'll be exposing purified samples to duckweed and see how it affects their reproduction. So the ultrasound process is just as simple. However, it's impossible to obtain an actual ultrasound detoxification machine for obvious reasons. But luckily, Mr. T devised a genius plan and yet again saved the day. It's said that a humidifier consists of an ultrasonic transducer. A transducer is basically a device that converts a signal in one form of energy to another form of energy, in this case into ultrasound waves. So after exposing it to a transducer for 5 to 7 minutes, I will perform the same test that I mentioned in the previous slide. Variables. Okay, so my independent variable is obviously the type of purification process used, either ultrasound or sulfidation, which makes the dependent variables these 4 things. First, the effect effect on E. coli, for which I'll be exposing the purified samples to E. coli and see how they react. Second, the concentration of the ions after purification, for which I'll be using the GCMS. Third is the effect on planaria, and finally the effect of duckweed. And I'll explain both of these processes in the experimental design. So that brings me to my hypothesis, and since there are certain compounds that either cannot be broken down by sulfide ions or require extremely high and unsafe concentrations of sulfide ions, I believe ultrasound will serve as a more efficient and safer way for detoxification. Now, water is something that you could fix with the device, but behavior around water is not something that just snaps into place. Hence, it is very essential to educate people about the adverse effects that their actions are having on them. And let's not forget that it is our own duty to save our planet. And I'm sorry, I just had to include the next song.